evening. Indeed, we're very fortunate, and thank you very much indeed, Sir Paul, for giving up your time this evening.
Michelle, please call me Paul. I don't.
Okay.
Thank you.
Oh, thank you, don't stand on ceremony. Thank you very much. Well, you've got an amazing CV. So Paul is a geneticist and cell biologist, and his major work has been on the cyclin-dependent protein kinases and how they regulate cell reproduction. And Paul is the director of the Francis Crick Institute and has served as president of the Royal Society, chief executive of Cancer Research UK, and president of the Rockefeller University. And he shared the 2001 Nobel Prize in Physiology or Medicine and has received numerous awards and prizes. And he was knighted by the Queen in 1999 and received the Legion d'Honneur in 2003 and the Order of the Rising Sun from Japan in 2018. He served as 15 years on the Council of Science and Technology, advising the Prime Minister and Cabinet, and is presently a Chief Scientific Advisor for the European Union. He has more than 60 honorary degrees and fellowships and is Chancellor of the University of Bristol from 2017. So great pleasure to welcome you this evening.
Well,
and
pleasure to
yes. Be. And if I may, I'm going to kick off the questioning. Now, you actually, your formative years were in Lion Park Primary School, I think, and also at Harrow County Boys School. But you know, I've got an interest in Harrow County Boys School in a number of ways, actually. And do you think in those particular schools that there are any influences, or did this come in your university career?
Well, you're absolutely right. And the first thing I wanted to say is that it's a pleasure to be speaking to everyone, because I come from that part of London, or at least I was brought up there. And Rachel contacted me from Kenton, because when I was at Harrow County Schoolboy, when I was at Harrow County School as a schoolboy, I was actually, my house was Kenton. And it suddenly all came back to me, because that's in fact what it was called. I went to primary school in Alperton, Wembley. I lived near the Ace Cafe, which some may remember on the North Circular there. I was on the road that led off at the back of the Ace Cafe, and went to the grammar school, you quite rightly say, at Harrow County School. And my school, Lion Park, was not a particularly academic primary school. Not so many people went to grammar school, only half a dozen or something of that sort. And I got into Harrow County School, and you mentioned, did anybody really influence me? And I think the person who sort of influenced me most probably was Keith Neal, who was the biology teacher. And I mean, he seemed, of course, old because he was a teacher, but he was only about 10 years older than I was. He was in his 20s. It was his first job. And what was fantastic about him, not only could he explain biology extremely well, but he was very, very interested in natural history. And being outdoors in the natural world is really the natural laboratory for a biologist. And we did all sorts of experiments, counting the insects, looking at badgers, and doing all sorts of other things, which were entirely in Keith's spare time. And it had really a great influence on me, growing fish, looking at them developing, and everything you could possibly imagine. The other person who influenced me was Ron Avery. Now, Ron Avery was the headmaster. We had a rather fierce headmaster before him, who was much less pleasant or much less easy to talk to. But Ron was a really humanizing influence. He moved from Harrow County School to Bristol, where he still lives, and I'm actually in contact with him. He's now well into his 90s. And he really tried to help me. I left actually the school a year early, and he was very worried about that, and because he was worried that I was not going to go on to university, which I did do, but he was very, very supportive of me, because coming from a non-academic family, it was all really rather strange to stay on at school beyond 15 and then to go on to university. So he thought that that perhaps that was a difficult decision for my family, and which it wasn't actually, but he was very concerned about me. Very, very nice man. And I thank both Keith and Ron for that early start.
Yeah. Well, I had the great pleasure of meeting them, because when I was part-time at Harrow County Boys' School in the music department, Mr. Avery gave me my first job, so I'm very grateful. It was rather strange, as there were very few female staff then. So
absolutely, very
yeah.
few. Yeah.
Yeah. And I'm going to pass over to Roy with his first question then. Got to unmute yourself, Roy. Do you want to unmute yourself, Roy? Sorry, but
yeah, you've got my questions there. But
yes.
The first one that springs to mind is, what do you do in your spare time, if you have any? What would you do?
Could you, I'm a little deaf, would you mind, Rachel, could you mind just repeating it so I can?
Yes. Well, Roy would like to ask what you do in your spare time, because
yes,
you've got such a full career.
If you ask my wife, she'd say I don't have any spare time, but I do quite a number of things. In fact, I've got quite a lot of interest. I mentioned natural history. I do go for walks in walks and in a very amateur sort of way, look at birds and insects and butterflies and the like, and plants. I was doing it. It's a beautiful weekend, and my house is in Oxford. I'm in London at my flat here tonight, and we went for a great walk along the Thames. And so I'm interested in natural history, but I also like the performing arts, theatre and classical music and opera, particularly theatre. And indeed, at school, I was in the Amateur Dramatical Society, which I very much enjoyed. But I'm additionally a pilot, actually, both a glider pilot and a power pilot of old vintage aeroplanes. And although I'm a bit more careful now than I perhaps was some years ago, and so I tend to fly with somebody else more now than by myself. But I'm still quite an active glider pilot. And until a couple of years ago, was going to the Alps and the Pyrenees most summers to fly in the mountains there. So those are some of the things that I do. I also, of course, have children and grandchildren who take up time as well. I'm not sure they're hobbies exactly, but they are certainly our interests.
Yeah, thanks very much. Ruth Toffer, would you like to go ahead with your first question?
Yes, thank you. I believe you were critical of the government in the early handling of the pandemic.
Yeah.
Now, it's very difficult to navigate a situation that hasn't arisen previously. But what would you have done differently? Well,
the first thing is to say that it is very difficult to do. But when you have something like this, a viral pandemic, the first thing to realize is that you don't know very much about it. And the only defense you have is really to trace it, to know where it is, to know where your enemy is, and then to put social distancing in place so it doesn't spread. Later, you can possibly develop treatments and hopefully develop a vaccine. And of course, we've seen in recent months the much more effective rollout of the vaccine. Where the government made, in my view, a huge error, and they really were big errors, was over testing, because they did not think carefully enough, and it was not just the politicians, it was advisors too, about how to set up rapidly effective testing. And they also did not understand how this virus was spreading. Now, what we did, I run a research institute, quite a big one. It's got 1,500 people working there. It's in next to St. Pancras. And we're a research laboratory. We don't do diagnostics. We don't do testing. But very early on in March, when we realized the extent of the pandemic would be, we realized that critically you needed to do two things: set up testing and protect the most vulnerable. That is those in hospitals and those in care homes. So what I did is I converted, and when I say I of course didn't do anything very much myself, but I had fantastic colleagues who converted part of the building into a very effective testing facility. And within 2 weeks, we were testing 2,000 people a day, with 24 hours turnaround, often 12 hours, and were providing protection for 10 North London hospitals and 150 care homes. I wrote to Downing Street and to the Department of Health, to the Minister, explaining that this was a very rapid way to get something working, because what we did is bolted our very efficient laboratory facilities onto pre-existing NHS and Public Health England logistics, so that this was protecting hospitals. You didn't have to, it was local, and you didn't have to set the whole thing up. And what they had decided to do was to set up mega labs, half a dozen or more throughout the country, but to do it from scratch. So they had very foolishly thought they could set up a massive facility sufficiently rapidly and connect it logistically and get the information back so it could help the pandemic. Well, it didn't do anything of the sort. In fact, the government testing was barely operational throughout the entire first pandemic spike, the first wave, whereas we were operating, as I said, within a couple of weeks. Now, I wrote to the Prime Minister, to Number 10, and also to Matt Hancock, and said we could roll this out across the country within weeks, and we could have 100,000 testing in the same way within weeks. And I didn't get an answer. I sent several letters ever, in fact, for 3 or 4 months. And I am a Nobel laureate and do run the biggest institute in the country. So I think it's disgraceful that they didn't take any notice of that. The second thing that we did, and I did it with Peter Ratcliffe, who's my director of clinical research, who has also won a Nobel Prize in medicine, so there were 2. So this time I tried to double the numbers. And what we had told them, and this time in the first week in April, was that nearly 50% of healthcare workers in hospitals in London were infected and were infecting their patients. And that half of those people had no symptoms. And at the time, the NHS were only testing, had limited testing capacity because they relied on the mega labs that weren't operating. And they did not provide the protection. And in fact, they said that people without symptoms shouldn't be tested when half of them were infected. And this led to rampant infection and to deaths, of course, in those who were in hospital catching the COVID, those who were vulnerable, and those who were sent back to the care homes, infecting all the care homes. I mean, this was a disgrace, what's happened. It hasn't yet been fully revealed. And that's why I was so highly critical. Now, it's not that these big mega labs, I should say, it's not that they shouldn't have been set up, but they would have taken 3 to 6 months to get going, which they did, and they haven't worked that effectively, I'm afraid, but there's big testing capacity now. It was simply that they needed to put local facilities in place. And remember, the universities and medical school laboratories were all closed. And I ran our one with volunteers. We had 300 volunteers working for nothing on this as a civil duty, And all of this was ignored, and nobody admitted until, in fact, a month ago in government, that they realized that people who weren't infected, without symptoms, could be infected. And yet we told them in April. I mean, it's been appalling. Much better with the vaccine, of course, run by the NHS essentially, and using vaccines developed by the scientists. But the testing was a fiasco, top to bottom. I'm sorry if I sound so clear about it.
No, not at all.
It really, when it's revealed, I think it'll show that we, and don't forget, we have the highest number of deaths per population in the world. Now, I don't believe the figures out of Russia, Brazil, or India for that matter, which will be probably higher. But of the nations who you can believe, we're still the highest death rate. So it just shows that we didn't get it right.
Ruth, I think it's very appropriate to ask your second question, actually.
Thank you. A few years ago, I believe there was a study or rehearsal of what would happen if there was a pandemic. And I've heard on the news that this wasn't really wasn't used.
Yeah.
Do you think any of the strategies suggested when the possibility of a pandemic was thought of were implemented?
Well, I have to admit, I'm not an expert in this area. I may have sounded like one when I was just saying what I did is I learned it on the job, to be quite honest. But what you're saying is completely correct. 3 years ago, I think it was called Cygnus. They did a sort of mock exercise as to what would happen if we had an influenza pandemic. And that revealed that we were utterly unprepared. But for some reason, it was never properly analyzed and not introduced. But it was well known, and even I knew about it, that that we weren't prepared for a pandemic, and there was a constant conversations going on about how we needed to do something, how we needed to have good protection clothing, how we needed to have vaccine capacity in the country, and how we needed to have plans for testing. All of this was known, but I think that the way that healthcare was being talked about too much was a bit like how you'd run a supermarket. You know, that you would have supplies coming in, and you'd only have a small amount of stock in your cornflake packets on the shelf, so to speak. And nobody thought, well, if you have a real pandemic, suddenly your supply lines are just going to collapse, which is of course what happened. And you can't operate like that. And I think I don't quite know the reason for this, but it's probably that if you put in the business consultants into such a situation, they will think, well, if you can just order it all the time and get it delivered within 24 hours, you save money because you don't have to have stores and all these sorts of things. So you can save relatively small sums, but a couple of percent perhaps. But what they didn't build into it was the need for resilience. And we should have learnt that in the trial that you talked about 3 years ago. I think we've learnt a lot of lessons over this. I should emphasize, although I've been critical of the government, it was a difficult thing to do, and we see many governments didn't do well. European Union was very slow on ordering vaccine. The US under President Trump was doing nothing, basically. And, these advanced nations like ours almost none of us were in a good state to deal with it. The Far East did better. And that's an interesting thing to analyze. One is that they've had more experience of these sorts of things, so they took the threats more seriously. And we see that in a number of countries. I mean, China's done well. South Korea, countries like Taiwan. And I think we got to turn to them and learn some lessons from them, which I'm sure we will do now, because nobody's not going to take this seriously in the future. At least I don't think so. I don't know if that answers your question or it was too long, but it's not that I know too much about that particular study, but I have heard enough to know that it wasn't taken notice of.
Yeah, thanks very much. Barry Kruger, would you like to ask your first question?
Yes, thanks. Was delaying the second jab of the Pfizer and AstraZeneca by 12 weeks a good decision, and focusing more on doing as many first jabs as possible for over 70s?
Yeah. Well,
I'd be interested in your view, having read so many different reviews.
Yes, it's actually quite a difficult call. And I think it was quite a brave call to introduce it, because in this situation, you had to be bold. And there was, in some sense, a lack of boldness going on in decision making. And that was the problem with the European Union, for sure. 12 weeks, I think, put it a bit too far. 3 weeks was what was recommended, what had been tested. We had no clinical data beyond 3 weeks from the trials. So it was a bit of a punt, quite honestly, to extend it to 12 weeks. I think most people would have been happy with 6 to 8 weeks or something like that, but 12 weeks was rather too long. In reality, it hasn't been 12 weeks in many places. I've had my vaccine jab and I only had to wait 9 weeks, and it wasn't that it was special, it was just what was happening. So I think there was some nervousness that 12 weeks might be too long. Now, was it wise? Well, I think it was wise to protect as much as we could those above the age of 60 or 70. And it did get those numbers, up more quickly. I think when we do the analyses, as we're now doing, because we're getting the information, it will probably reveal that the protection from the first jab began to go off before the second one. So I suspect that will come out, but this time I don't criticize the decision to do it. I think that it was a very difficult one, and it was not an unsensible thing to do. So it may be in the end there will be a bit of criticism of it, but I don't think in this case it will be justified. Quite different from what I said about testing. Quite different.
Thank you.
We had another question from Roy. Is why have the Europeans been left behind with their COVID vaccinations?
Yeah, well, I know a bit about this actually.
Yeah.
I've been, worked as an advisor. I've just stopped being an advisor, or I will do in a week's time actually.
I see.
I've done it for four years. It's due to something called the precautionary principle mainly, I would say. The precautionary principle is a principle that is which was talked about quite a lot 20 years ago. And it's really that it sounds sensible, but is not sensible in all circumstances. And what it really says is this, is that if something new is happening, particularly if it involves science and technology, then until you're sure that that whatever you're doing is safe, then the precautionary approach should be not to introduce it. And this arose, I think, partly due to thalidomide. I mean, many of you, I think, are probably of an age to remember the thalidomide crisis, where a drug wasn't fully tested, and that led to catastrophic problems in pregnancies and the babies that were born. And as a consequence of that, there was a sort of an attitude where you only did something if you had all the data and all the evidence to make sure that it was safe and the proper thing to do. Mm-hmm. Now, in a sense, how could you argue against that? But what happened with the European Union is that they didn't want to adopt a vaccine until they were sure that it worked and it was safe. In other words, they relied on the evidence. And at first sight, that seems sensible, but in a way, it would have been a sensible decision in what I call peacetime, but not a sensible decision in what was the case, which was wartime, really. Mm. We were faced with very difficult decisions that required bold action using what data you could have at the moment. And therefore, in the UK, under the leadership of Kate Bingham, who some of you may have read about. Now, she's one of my trustees at the Crick, so she's one of my bosses. I have quite a few bosses there, but she's one of my bosses. And she did a fantastic job in evaluating the vaccines that were coming along and making a hunch, a guess, as to which ones were likely to be most effective. And she did it well, and she got it right. So the vaccines that we bought, the two novel ones, which are Moderna and the BioNTech Pfizer one, and the more conventional one, which came out of Oxford and AstraZeneca. So the bold decision taken by Kate to buy those vaccines early and to get them coming early was very, very good. In the European Union, they hung on waiting for evidence and then got themselves too low down in the queue. And it's because and that you saw other aspects of it as well. You saw Macron, for example, saying that the vaccine, the AstraZeneca one, shouldn't be used for those over 65. I don't know if you remember that early on. And that was because there was no clinical evidence that was statistically significant that it worked for those over 65. So it wasn't that he was following the evidence. It was just so very likely that it would work. It just wasn't that the evidence was there. And because it worked for younger people, and there's no reason from what we understand about vaccines that it wouldn't work. But he was being advised or guided by saying there's not evidence for this, so it would be unsafe to roll it out. Of course, it's in a country which is rather an anti-vaccine country, so he may have been worried also about that. So where do we come out on all of this? It's really that, of course, we have to rely on evidence, but sometimes it's more dangerous not to do something than to do it. And in this case, that's a judgment call, in some respects, a political judgment call. And if you have a culture which is risk averse and follows the precautionary principle, when the going gets really rough, they're not going to make the most sensible decisions. And that would be my analysis as to why continental Europe hasn't done so well. It was also slow because they made a decision that all the countries should work together. They didn't have to make that decision. It wasn't forced upon them. And in fact, there were some countries who argued to go alone. But getting 27 nations to make a single decision quickly isn't easy. And they took 6 or 8 weeks to make the decision as well. So several factors influenced why it was so slow.
Thanks. So your role is finishing. Is it because of the term of office is coming to an end, or?
Yes, it is. It's a term of office that comes to the end. The UK has, of course, left the European Union, but they can take advisors from around the world. And of course, we were in the European Union when I was appointed 4 years ago. And it's a term, and it finishes in a couple of weeks' time. Yeah.
Would you like to say a bit more about that and how your work's developed?
Yes, I can. I've advised both the UK government for 15 years on an equivalent committee called the Council for Science and Technology, so on a number of prime ministers. And that is an advisory committee which is chaired by the chief government scientist, who would be Patrick Vallance at the moment. But the system that operates in the UK has a single chief government scientist. In the European Union, of course, it's much larger. They have 7, so I'm one of 7. And that allows you to have both a greater breadth of skill set and discipline expertise, biological, physical sciences, medical sciences, informatics, and so on, than you can get with a single person. And it also allows a spread across the whole continent of Europe, because from the east to the west. When I joined it, was rather in favor of the system that we had in the UK, the single person. But having worked in it for 4 or 5 years, I actually think this is superior, certainly in the continental European context, because it spreads the load of decision making across more people. The problem with it is the one that I've already described, that means it can be a bit slower, whereas a chief government scientist can move fast, but the risk of an error is perhaps greater. So there's pros and cons either way. My experience is that there is something to be learned from it. But if you have a crisis, it's very good if the people making the decisions are few, because you're more likely to come to
some agreement. Yes, I understand that. Yes, Barry, you'd like to get to your second question? That fits in very well.
Yeah, thanks, Rochelle. Some countries are only using AstraZeneca for people over 30. So people in third world countries have a high number of people over 30 and under 30.
Under 30, yeah.
I've seen quoted under 30 in India around 65%. If limited to over 30s, would this have a much slower impact in terms of trying to achieve a reasonable rate of inoculation in the poorer countries like India and third world countries?
Well, this is another very difficult problem. It's full of difficult problems, this
absolutely.
pandemic, of course. And I'll give you my commentary on it. The reason for the concern about younger people and the AstraZeneca is these unusual blood clots that occur in the brain. Now, they are very rare, but they are not absent. I mean, they do occur, and it is linked to the AstraZeneca vaccine. And there have been deaths, I mean, a number of deaths as a consequence. So it is certainly safer in that regard not to use that vaccine for the those under the age of 30, some say under the age of 40. However, there's a couple of things I can say about it. The numbers who suffer this are a lot less than those women who are on the pill would suffer similar types of blood clots. I mean, it's a different type of it. But so and yet, even though I may not quite know the figures, forgive me for that, but I think it's 5 to 10 times more common on the contraceptive pill. Yet that is a risk which people will accept. But there does seem to be some nervousness about doing it in the case of a vaccine, but it is clearly quite low. And the other point to remember that is the one you're mentioning is that not using AstraZeneca for young people could have, particularly in poorer countries, an impact on giving protection to the whole country, because the AstraZeneca vaccine is cheap compared to the Pfizer one. It's either 10 or 20% of the total cost, so it's a lot cheaper. And it also, as we all know, easier to distribute. And the Pfizer vaccine has to be kept at -70 degrees centigrade, whereas the AstraZeneca one is okay in a normal standard deep freeze that you would have in your kitchen or close to that. So it's much easier to use AstraZeneca in the developing world in a India-like situation. And the lives that would be lost due to the blood clots are significantly less than the lives that will be lost by the fact that individuals are not vaccinated and therefore will catch the COVID and therefore die. Some will die because of that. So it's a lot to do with the probabilities, which is very theoretical, of what happens if you take it and what will happen as a population as a whole if you don't take it. And this is what's got to be factored in when the public health people decide what to do. If you're asking what I would do, I would deploy the AstraZeneca vaccine for those under 30 for sure in those countries such as India, because I think we see what's happening now. There's various reasons why this is happening. The vaccine rollout was slow, and then there were all these mass rallies, both religious and political, which have spread it terribly. And so we're certainly getting hundreds of thousands of new infections every day, and it may go up to 500,000. So I would get the AstraZeneca vaccine out there and give protection to the whole population. So that's a decision I would make. But it is difficult. I mean, if you were, if you had a 30-year-old, if you were a 30-year-old, they will think about these things. And there's a lot to do with sensible communication and trust in your political leadership and advice that you're getting from medical advisors, like our chief medical officer, for example. And if people don't have trust in the system, then they're going to start believing the sorts of things that Mr. Trump said, like drinking disinfectant might help you, for example. So we're learning something about political trust, I would say, as well, in the present situation.
Thank you very much for the answers to my two short questions. Thanks very much.
Sorry, am I talking too much?
No, not at all.
No, very interesting, very interesting.
Well, before we pass on to some other topics, I'd like to know just about the role of the World Health Organization. And there's a great furore at the beginning of the first lockdown, but they seem to be rather reticent in the last couple of months. So.
Yes, I don't know too much about the WHO. I'll be quite honest with you. I'm not actually a medical doctor. I'm a PhD. So yeah. It's, again, something I've learned on the job a bit. I was asked to become an advisor for the WHO a month or two ago, which I didn't, because I didn't think I was expert enough, and I also have a lot to do. They were a bit slow on the uptake, I think, at the beginning, to get going with it. After that, I think it went quite a lot better. WHO is very important, I've got to say, but it's yet another one of these complicated organizations. It's 130 countries, I think it is, that are members of it, and getting decisions made is very slow there as well. So on the whole, it's quite a good thing, but it was slow in this particular case. At the Crick, I should say, we have a WHO center for influenza. There's 5 in the world, and we have one of them which monitors the spread of influenza and the virus, the type of influenza virus that is current in that season. So we have some experience with that, and indeed I'm going to try and see whether we can put in place a more general viral surveillance facility at the Crick, which would include COVID and other viruses, building on our experience with influenza. So I think we need something like the WHO. It needs to be improved, but it's not a disaster by any stretch of the imagination, but it was slow in getting going at the beginning of the crisis.
Yeah, definitely. Thanks very much. Mel, would you like to ask your question about the Crick Institute?
Good evening, Sir Paul. It was just a thought about the cost of all this research. Yeah. The Crick Institute, at the time, I believe, it was costing several hundred millions to construct and get going, and there's a very high annual running cost. How do you explain these sort of figures about the cost of medical research?
Well, the short answer is it's very expensive doing medical research. I mean, there's and in the academic sector, which we're in, the not-for-profit, it's significantly cheaper than it is in the private sector, for example, where it costs a lot more because they pay much higher salaries. The institute itself, you're right, it costs 450 million to put up. It, however, merged 3 previously existing institutes, which were at Mill Hill, the National Institute of Medical Research, and then the CRUK London Research Institute, which was in Lincoln's Inn Fields, which I was once director of, actually, and a second laboratory there in Mill Hill. Now, although it was 450 million, actually the land for those that was sold from those 3 sites contributed easily a quarter of it. I don't no longer have the figures exactly in my head, but about a quarter, in fact more than a quarter. And CRUK raised 150 million from donors over a couple of years, and then we got money from the government through the Medical Research Council. Now, the reasons these are expensive buildings is that if you've ever seen the Crick, I don't know if you've ever seen it from outside, but it's a highly sophisticated building. We have very very strong containment facilities so that one can work on viruses and so on. We have very sophisticated equipment and so on for carrying out quite sophisticated techniques. Now, the cost of it, we have 1,500 people working there, and the cost in total is about 150 million pounds per annum. So this is again a lot of money, although is about 2% of the expense per year that the country spends on public spending for research activities. So it's not it's small in comparison of putting if you put everything together. And to give you a comparison for that, University College London, that you might be familiar with, the big university, that has 1,000 research groups, and we have 120, so it's 10 times bigger than we are. And that costs a lot of money too. I'm afraid we just have to accept it's expensive. But putting all of this together, the public funding is less than half of what is spent in private industry in supporting these activities. And the salaries and so on of my colleagues are low. I mean, in America, they'd be paid several times more, has to be said. And I don't think we should never forget they do see it as a public good. I mean, it's not that they're paid low sums of money. I mean, you know, they're paid more than nurses and so on, but they are very highly trained and very international, so they could go anywhere in the world and get paid more. So in many places, so it's just an expensive business. But then if you look at it, look at the amount of money we've just spent on the pandemic, when we could have been quite a lot better prepared with the right research. And I have a feeling that it would be right to make sure we're better protected by understanding things better. But then I would say all of this, I do run the damn place after all.
Thanks very much.
Thank you.
Can you explain to us what the eukaryotic cell is?
Yes, I can.
Yeah, thank you.
We are eukaryotes, and I'll tell you exactly what it is. There's actually 3, we'll call it 2, make it simple. 2 basic kingdoms in the living world, what are called prokaryotes and eukaryotes. Now, karyot is Greek for nut, actually, and or something like that. And I did Greek at Harrow County School, but I was at the bottom of the class, so I'm never confident of my...
What, with Ken Waller, was it?
Sorry?
With Ken Waller?
Yes, he was there. Yes,
yeah.
That's absolutely right. But I was terrible at it. Anyway, and what that describes is the nucleus. Eukaryotic cells, which range everything from yeast, which is what I do my research on, through to human beings, which are the most sophisticated, at least we like to think we are. Every cell in our body has a nucleus, and you see that, and it looked to the early microscopists like some sort of nut in the cell. The prokaryotes are bacteria-like, and bacteria do not have a nucleus, so you can't see that in bacteria. So those are the 2 kingdoms: eukaryotes, of which we are one, because we have a nucleus in our cells; prokaryotes, which are bacteria, which don't.
And could you explain to us what are cyclin-dependent protein kinases?
Yes, cyclin-dependent protein kinases. I've been working on these for a very long time. I was one of the people who discovered them. I'll tell you what they do. I tell you what they do chemically, and then I'll tell you what they do for eukaryotic cells, okay, what their function is. So cyclin-dependent protein kinases, they're enzymes, and enzymes catalyze chemical reactions. Now, what protein kinases do is they transfer a phosphate molecule onto other proteins. That's their catalytic activity. And if you put a phosphate molecule into a protein, a phosphate molecule is quite bulky and it's highly negatively charged. And a consequence of that negative charge is that the protein that gets phosphorylated, which gets a phosphate put on it, changes its structure and changes its function. So protein kinases are used as very rapid switches in a cell because they change very rapidly the shape and properties of many, many proteins. And so if you want to switch behavior in a cell, then a protein kinase is a good way to do it. The cyclin-dependent protein kinases, which is the family that my lab discovered, is one where there is the protein kinase is in two bits. One which is the protein kinase, and then there's an activating other protein that sort of joins together with it, which is called cyclin. So it's cyclin-dependent kinase, because this is needed for this to work. Now, what does it do? Well, what it does is the cyclin-dependent kinase phosphorylates many hundreds of different proteins in the cell. And these proteins control the reproduction of a cell from 1 to 2 cells. Now, when a cell reproduces, it's born and it then has to prepare itself for the division process at the end of the cell cycle. And a number of things at the end of its growth cycle. And there's a number of things that have to happen there, including critically, you have to copy your DNA, and then you have to separate the DNA at division, otherwise the genes are not put into each cell. And that takes a program of work. And that program of work is controlled by the cyclin-dependent kinases, which phosphorylate one set of proteins at the beginning of the reproductive process that start the process off. And then as its activity increases, which it does, it changes the function of more and more proteins until eventually it causes the cell to divide into 2. So what these enzymes do, I call them CDKs because that's much easier to say than cyclin-dependent protein kinase. What these CDKs do is control the reproduction of all eukaryotic cells. So every cell in your body, and 1 billion of them has probably divided since we started talking, is a consequence of this particular cyclin-dependent kinase. Now, my lab, and there was another one in America, discovered these in the 1970s and 80s, so a long time ago. And we discovered them in yeast, which is what I work on and what my colleague did in America worked on, somewhat different yeast. And we showed they controlled the reproduction of yeast cells. And then my lab went on to show that the same enzymes exist in human cells. And I just want to tell you about that experiment if I'm not talking too much, because it is a very nice experiment. It was before we had any human sequences, human genome sequence. So this was 10 years before that. And what we had was a yeast cell that couldn't divide because it was defective in this enzyme. And my lab reasoned that maybe humans had the same gene. Okay?
Oh, I see, yeah.
And so what we did is we took a gene library from human cells and metaphorically sprinkled those genes on these defective yeast cells. And what I'd reasoned was that if the if us humans had a similar gene and it was taken up by the yeast cell, and it could perform the same function as the yeast cell, then that yeast cell, defective in the CDK, would then be able to reproduce and grow and divide. This was a crazy experiment if you think about it, because yeasts are so simple compared with humans.
Yeah.
We of course diverged 1,500 million years ago, roughly. So what this means is, and that experiment worked. I mean, it had no right to work, but it did. And what that showed was that humans and yeast division is controlled by exactly the same gene. It's extraordinary. And not only that, but it's been working like that for 1,500 million years. 1,500 million years. Now, just to put that in context, dinosaurs went extinct about 65 million years ago. I mean, a much, much shorter period of time. And so this whole system is extraordinarily conserved. And every living thing you can see around us. I mean, I could show you my, look at my plant over here, all the cells there are also controlled by the same enzyme. We isolate it from plants from everywhere. So it's very important for the growth of our own cells and also of every other living thing that we can see except bacteria on the planet. And that's important for the growth and development of all living things, and of course goes wrong in cancer. The genes get activated to cause reproduction when certain defects occur in cancer. Yeah. So that's what I've spent my life studying. In 5 minutes, you've got it there.
Yeah. Thank you very much indeed. And Ruth, would you like to ask your question?
Good evening, Sir Paul. I'm going to start with a statement. I am very upset about Brexit. I wanted to remain. But what I'd like to ask is, what effect is it going to have on scientific research? And in particular, if we can't exchange students and scientific researchers and Nobel Prize winners, how on earth are we going to progress?
Well, I was also not in favour of Brexit. I should say that. It's not that I think the European Union is perfect. It absolutely isn't. But it is our nearest neighbours, and it is the way who have very common ways of working. And I now ask, who are our friends really? And so nothing is ever perfect. But I'm not even sure where we're looking for friends. But you were asking particularly about science. Well, one thing, and I have to say, I fought very hard for this, was that that in principle, the government has agreed that scientific research will remain closely linked to the European Union, and the funding mechanisms and the way of working in principle, and I'll say why I'm using that word, could remain and could be in place. And in fact, in my institute, we are allowed still to apply to the European Union funding agencies. And this very week, 4 of my researchers won £10 million worth of grants. We've been learning that it's from the European Union. So that means it may not be as bad as it might have been. Okay, for science, just to allay your fears. However, it does depend upon the UK putting money in for the research purposes, and they didn't put it in the budget. They didn't put it in the budget, and as a consequence of that, I've had quite a big fight to make them transfer the money into the budget, which was successfully done. And you may have read it in the papers. It didn't really get to the top about 3 weeks ago. And I had to do quite a lot of writing of letters and so on about it. And others, of course, did as well. And persuading people to come to the Institute so we could discuss it with them. So if we can keep that going, I think we'll keep the science going. And that, I think, is a very important part of this. And so the point you were making about how important science is critical the future. And the European overall science activity is leading in the world. It's beginning to overtake the United States. And we see it rising in China and the Far East. And we're in danger of separating ourselves from it. But I'm hopeful that that won't be the case. But we must be vigilant, and we have to keep on their backs. So, I mean, we've only rescued it for 1 year. So we have to see what happens next year.
Does that mean there's a free flow of scientists and students?
At this moment, scientists, there's a problem with student flow because there was a scheme called Erasmus. I don't know if you've known about it. That didn't survive. So that was one thing that didn't. Research did, students didn't. Big mistake again. I would say that there's been a substitute program put in place, but it isn't the same. It actually funds our students to go elsewhere, but not students to come to the UK. And what you need to know about science is that the relationships you make when you're young persist for the rest of your lives. And international exchange at that time is very important for the future of research later. I mean, many people I now talk with and collaborate are people I met when I was very young, just by the nature of being a human being, really.
Thank you.
Thanks very much. Irene's got a question about schools. Go ahead, Irene.
Oh, good evening. Thank you. You have said that in England, we don't learn how to discuss science. I'm not quite sure I understand what you mean by that in terms of we learn about science. So how do we learn about discussing it? And how do you think that schools should tackle that?
Well, I think what I was trying to say really was that we shouldn't learn science as just a series of facts about the world. That's important, but you also have to understand how science works to bring about knowledge of the world that leads to those facts. So and that's what perhaps when you said how to discuss it, it's being able to know how science works and to know the process, because otherwise, if you don't know how science works, it's very difficult to know when you should believe what somebody says and when you shouldn't believe what they say. So again, back to the poor old Mr. Trump saying that, you know, drinking disinfectant will cure COVID for you. Of course, it's not reliant upon any science at all. But if the President of the United States says it and a scientist says something different, you have to know what the basis of why he says that and somebody else doesn't. So that's part of this. But so that might have been what you were thinking of. Another thing might be something else, which is this, is our society is increasingly complicated and increasingly influenced by technology and science. I mean, almost everything we do. What we are doing now, talking on, what's this, Zoom? I think we're on Zoom, aren't we?
Yeah, we are.
And, you know, I'm sitting here with my Apple computer, and science and technology has produced all of this, and everything around us is influenced by it in so many decisions, you know, what do we do about energy and nuclear power, wind power? What do we do about medicines? What do we do about agriculture? Are all being influenced by quite sophisticated knowledge about how the world works. I mean, by which I mean plants, animals, the physical processes, and the impact that has on technologies and applications. And for a democracy to thrive, we as a society are going to have to embrace issues and decisions that have quite a big science element. And to be able to have an effective democracy, in my opinion, my view at least, we need to engage and discuss scientific matters at a level which at least allows us to come to decisions about what we should do, like should we have any nuclear power, for example. Now, I mean, that's because it's cleaner, in terms of CO2, but obviously has drawbacks. And this sort of decision making is critical for our future. And we have to find out ways of how we can discuss it if our democracy is going to be a healthy one. Otherwise, the populists will just say whatever they like, or other countries which don't respect democracy so much will just sort of enforce things top down. And so I think it's a critical for the 21st century, to be quite honest. So that's two aspects to perhaps your question.
Thanks very much. And we had a question from Roy asking about what is your opinion about the origination of the virus?
Well, I'm no expert again, but
no.
My friends who are say that by far, and I have one or two researchers in the Institute who really know about this stuff, say by far the most likely origin is that it had its origins in a bat, and it then went from a bat to another animal, probably a pangolin, and from there it hopped into humans. And the reason they say that is that they can study the evolution. You know this spike protein, which the vaccine is sort of made against? You can look at the evolution of that spike protein in different viruses and trace where it came from. There's very little support for the idea that it escaped from a laboratory. The experts that I speak to say that's extremely unlikely, but the trace of how it happened, did it go through that market in Wuhan?
Yes.
That doesn't, it's not really clear that that's quite what happened. And I'm not entirely sure we're ever going to find out. There's of course lots of conspiracy theories around. I mean, and there will be, but I'm sure for a while. But for some reason, I don't fully understand. Quite a number of these viruses do seem to come from bats, and I just simply don't understand why that is the case. Maybe somebody does, but I don't.
Yeah, thanks. Now we've got a genetic question for you from Robert Brody.
Hi, good evening, Sir Paul. It's so nice that you. We who live here know that Kenton's a magic word, and it's lovely to know that it still has its power. That when Rochelle waved it in front of you, it pulled you on board. So, welcome.
Thank you very much.
When I first heard we were having a geneticist online, I can't imagine how it happened, but COVID wasn't the first thing that came to mind. And I don't know if my question is within your sphere of influence, but your talk about CDKs was maybe the segue. Now, the question is, traditional Orthodox Judaism seems to be strongly against any kind of conversion to Judaism that it's not under its own rabbinic authority.
Yeah.
Despite this, there are huge numbers of non-Orthodox conversions taking place around the world. And according to the Orthodox authorities, this is leading to a generation of non-authentic so-called Jews and division into two societies, one not accepted by the other. My question has two parts. Firstly, how authoritative and strong are the genetic links through the generations of Jews that link us to our biblical forebears? And secondly, how is it that genetic traits can accommodate that many Polish Jews look like Poles, Swedish Jews look like Swedes, Chinese Jews, Ethiopian Jews, Iraqi Jews, etc.? Can British Jews, mostly of European stock, have confidence in their inheritance?
Well, I think you've answered the question almost yourself, of course. I mean, the fact is, it's highly diluted from your forebears, the genetic contribution. And I would probably more focus on the cultural and religious traditions that are handed down that don't change, rather than the genetics, which tend to, and which, of course, often the different nationalities and different people with different religious faith often share many genes with neighbors who have quite different quite different religious beliefs, quite different cultural traditions. And so my focus, I think, would be on the cultural inheritance of a society and the religious inheritance, which is handed on not through your genes, but through the way you're brought up. So I think that's the way I would look at it. If you rely on genes themselves, they will be highly diluted since Abraham, for sure.
Thank you. That's very interesting. I'm not sure a lot of people in Orthodox Judaism would agree with you, but it's lovely to hear it. Thank you.
Yeah, thanks very much. Do we have any more questions for Paul before we hand over to David?
Well, I've got a question.
Yes, go ahead, Peter.
I understand that you've got a great interest in natural history, and particularly is it in beetles, that you're very interested in beetles. And I wondered if you could say something about that.
Well, I am interested in beetles. I'm a strange person, really. I mean, beetles are
really interested in green fly.
Beetles are amazing. They're amongst the most successful type of animal on the planet. I forget how many species there are, but there's probably 500,000 of them, something of that sort. We have nearly 5,000 in the UK. And they are wonderfly resilient sort of insects. You know, these hard, you've seen a beetle, so you know they have these hard, they're called elytra. What they are is they use 2 of their wings. If you look at a fly, it's got four wings, 2 on each side. And a beetle, usually, not always, but usually will turn over time in evolution. 2 of those wings turn into a very hard shell that sits over the beetle, protecting it. And when it wants to fly, it lifts that up and it has 2 more wings, which are normal wings. So this means, unlike a fly or most insects, dragonflies and so on, which are rather vulnerable all the time, beetles are like armor-plated, tanks that can fly. And there's something fascinating about beetles. I've got, I don't know if you can see behind me, this is a whole book of, this is a book published in the 19th century about German beetles. And they're full of wonderful pictures, because the Victorian 19th century entomologists would collect lots and lots of them, and including Charles Darwin, of course. And Charles Darwin famously was catching beetles, and he had one in one hand and one in the other, and he wanted to get a third. And so he put it in his mouth, one of the beetles in his mouth, and then grabbed the third beetle with his other hand. Now, when I was at Harrow County School, I used to collect insects. It's not allowed really now, we don't do it. And I still have an interest in beetles, and this is all my Darwin collection. These are all early editions, first or second edition, and my beetle ones are down here. So I just like them, really. I'm not even very scholarly about it, I'm an amateur astronomer too. I just like looking at the planets, and I don't try and do research or anything. I just look at it, and it's the same with the beetles. Does that explain anything?
Well, thank you very much, because I'm a professional entomologist, and I love insects.
So I'm teaching my grandmother to.
Oh no, no. I'm just interested to know that there are other people out there who are interested in insects too.
Well, they did make me an honorary fellow of the Entomological Society, Royal Entomological Society, because I was always waxing lyrical about insects.
Well, that's good, because the Royal Ensocs just had a 2-day wonderful meeting all about greenfly and aphids, and I took part in that, and it's really great fun.
Well, now
I can indulge that now I'm retired.
Well, you've embarrassed me now, because here was I telling you about elytra and.
Not at all. No. It's good for everybody else to hear it.
Well, I've just written a book actually called "What Is Life," and what flits through all the pages is a yellow brimstone butterfly, the harbinger of spring.
Yeah.
And I use it as a metaphor quite a lot through the book. I don't know if I've got a copy here. Maybe, hang on, I'll show you.
You've really brought him to life.
Yes, right.
So this is the book, and this is the tree of life, okay?
Right.
Yes.
But now if you look on the back, you'll see there's a brimstone yellow.
Oh,
Oh, I see.
right.
Yeah.
Okay, you've just persuaded me to buy a copy.
That's right. I know,
It's
and
very
all of us
cheap.
actually.
I said they had to sell it cheap, and it is cheap.
Thank you very much.
Thanks very much. Well, I'm sure your sales are going to go up now after this meeting. Do you have any more questions? Otherwise, we'll hand over to Dave. He's got something he'd like to show you.
Good evening, Paul.
Hi.
Can't hear you.
Yeah, can't hear
Can't
you.
hear
so-
Is that better? Can you hear me?
Yes,
okay?
that's great. Yeah,
Okay,
lovely.
okay, yeah. We had Sir Nigel Sheinwald a few weeks ago,
Oh yeah,
and Nigel is still in touch with Roy Avery as well,
he is indeed. I knew that. Yeah.
which is interesting. I didn't start Harrow County till 65, and I think you left 67.
Correct. You are
yeah.
correct.
And I wasn't in Kenton, I was in Northwick. But I did have Keith Neal, but that's not what I want to show you. I have a photo from 1963.
Oh yeah.
And I believe if it isn't you, then the whole thing's irrelevant. But I think I've got a photo of you perhaps getting your very first award in 1963.
What the Duke of Edinburgh's award or
No.
oh?
Let me see. Let's just share the screen.
Okay.
So here we go. Share screen. I would like, if you could, right, can you all see a photograph?
Yes.
Is that red arrow pointing to Paul Nurse?
Do you know it is? This is the mayor of Harrow, isn't it?
That is 1963, the mayor's parlour. Do you know what, can you remember what it was?
I got a prize from the mayor for something, but I can't remember what it was. This is amazing.
It is.
I love doing this. Okay, this is, I always bring things down to my level.
Your sound's gone a bit.
You still hear me?
That's better. Good. Yeah.
Okay, sorry, I will shout. Okay, this was a freedom from hunger competition. And you either had to do a poster or I believe write an essay.
I think I wrote an essay, I think.
Yeah, so I think, so if this was 1963, you would be in the 4th or 5th year at Harrow County?
I would have been 14. I think it must have been the 3rd or 4th form, I think. Anyway.
Yeah.
Okay, so so the question is, why have I had this photo in my photograph album since 1963? And the reason is, and I don't think I'm going to be able to move the arrow, no. If you look to the left of the mayor, the little boy in short trousers.
That's you, yeah.
That's a 9-year-old David Hantman who did a poster for Freedom from Hunger and won that age group. So that's...
David, is there any chance you could send a copy of that to me?
Absolutely, 100%. 100%.
Fantastic. I don't remember it very well, and I certainly don't remember. I had a terrible hairstyle, didn't I?
Yeah.
Oh yeah, but now then, I'm younger and you still got your hair, so obviously you listened to what Keith Neal was saying in the Bible.
The sound's gone a bit, David.
Hey, can you hear me again?
Yeah, great. Yeah.
Yeah, you mentioned you lived in Alperton behind the Ace Cafe.
Yeah.
Probably around Mount Pleasant.
Yes, around Mount Pleasant, exactly. Yeah.
Does Bilton Road mean anything to
Yes,
you?
it does.
Norman Hart optician.
Yes, it does.
My late father always claimed that you were one of his patients.
Well, that is possible. I mean, I don't really remember, but why do I remember the name of the road and the name of the shop? I mean, it's got to be something going on.
Well, yeah. But it was a long time ago. It was a long time ago.
Yeah.
Anyway, that was how we decided to end this. I'm going to pass you. I've been spotlighting everybody else in the background, so I'll get that photo to you with pleasure. And it's lovely.
That would be fantastic.
Rochelle, over to you.
Yeah, thanks very much, Lou. So what a wonderful end to the evening. That is astonishing. 58 years ago. So that's quite remarkable. So thank you very much, Paul, for being with us this evening. And we appreciate all the questions that have been asked in advance and this evening. And you've drawn the very best from you this evening. And it's been actually fascinating to learn and be educated by you on so many topics raised. So thank you so much. And a special thanks to our special tech team, Jane, David and Morris. Thank you very much indeed. Thank you so much for being with us.
Well, thank you. It's been a pleasure talking to you. And thank you for the invitation.
No, it's been wonderful. Thank you. Thank you very much.
Bye-bye.
Yeah, good night.
Enjoy the rest of the evening.
Yeah, thank you.
As is usual, I will leave this running for another 5-10 minutes if people